Today I'll be talking about perturbing histology with generative encoding and linear mixed forms. So the idea is to take a histology tile, perturb it by converting it to gene expression first, changing it in the gene expression space, and then changing it back to the histology space. So as you can see, soon the three images of the three tiles are all the same histology tile, but as you change different sets of genes, they go down a different differentiation route. So the idea of a bridging transformation is to take one modality, such as spatial histology, to another, such as RNA. Now, once this bridge is built, we can go back and forth from one space to another, and thus we can look at certain ideas and topics. Firstly, if we talk about developmental trajectory of tissues and cells, we could, for example, take the cells on an RNA velocity plot, convert them to histology, and then we can image the cells as they change and progress from one route and go down the path from in an RNA velocity plot, for example. Another idea is to look at tissue samples and how they develop over space and time. And this could be related to developmental tissues such as stem cells or organoids. Now, the next idea would be to look at gene dynamics or cell interactions. The ability to visualize gene expression in the histology space will definitely allow a more interpretable view of how genes affect histology, morphology, and how cell interactions on the morphology alter the gene expression. And this could extend databases through those concepts listed above. And then in the last one, we could look at how genetic variants finally map onto histology to influence the cell structure, shape, and tissue abundance. So here I have an image of a histology heat map, histology gene expression freeze. So here I have a histology tissue sample where I've taken each tile and converted to a gene expression. And in the gene expression space, I've taken the k nearest neighbors and cycled through each, say, the top 15 nearest neighbors, replacing each of the original tiles with those nearest neighbors in the gene expression space. And you can see that the gene expression space really does define the spatial histology space. So when I say define, I mean this mound here, the circular globular tissue colony, it is being defined by the gene expression within this area, and that is why the nearest neighbors in the gene expression space recapitulate the original image. And so you can imagine that in this area, in the dark, where the dark cells are, they'd be expressing high genes related to the nuclei. And then as you progress into the extracellular matrix, it would be expressing less of those genes and then back up again as it progresses over these cells to the left. Now, this is a closer snapshot of the image before, and you can definitely tell now that there are cells and the nearest neighbor tiles are moving forwards and cycling. So the idea is to build a bridge of transforms. That is a bridge across gene gradients, within gene gradients, and across spatial histology. And just from this image, you can start to tell that if you map them across from one gene to another to the histology space, we are building a multidimensional lattice of information. So how do we formulate the transform? So for example, if we had data sets Y, X, U, and W, where they could be any data set, how would we formulate the transform between Y and X? Well, it's quite a straightforward mapping with a function from function of X to Y and the inverse of that function from Y to X. So for example, as I mentioned before, this could be on histology and spatial transcripts. Another idea could be on methylation and chromatin, or the last one could be from the progressing along the central dogma of biology from genetics, epigenetics, transcriptomics, and proteomics. Now here with the linear model, I just want to state the simple terms that a function could exist in. This is quite a basic foundation, and so I'll just gloss over this quickly. So the function here is defined by the matrix beta, y and x are your data sets. So the beta parameter transforms x to y, and then the inverse of the beta matrix transforms y to x. Now note that this is a slow transform when x has many genes. So for example, 30,000, that will just become inhibitively slow when you're taking the inverse of a 30,000 by 38,000 matrix. But just notice that here, for example, if y is your histology and x is your gene expression, converting histology gene expression and then perturbing the gene expression space, and then converting the gene expression back to histology gets you a perturbed histology image. So that wraps up part one. And now I'll move on to part two. So the main method that I'd like to talk about is called a linear mixed form. This is very similar to a linear mixed model. The only difference is that Y can be a matrix of phenotypic covariates or response variables instead of a vector of phenotypes. And alpha is now added on to convert the matrix or vector into a larger or smaller subdimensional space based on how many phenotype response variables we have. And likewise, beta and U are now can also be matrices that convert all of the information Y, X, and Z into the same subdimensional space. So how do we convert them into the same space? Well, we want to decompose Y, X, and Z into a set of latent variables that are common or shared across all of the Y, X, and Z's. So here I state that Y is decomposed as K plus H and then matrix multiplied by alpha transpose, where alpha transpose is related to alpha in the top line, Y alpha. So that implies that Y alpha is equivalent to Y, or well, K plus H alpha transpose alpha, but I'll go into more details on that later. And then if we look at the next line, we have X is decomposed to K beta transpose. Now this K here is similar to the K in Y, except that in Y we also have this H latent parameter where the H appears in Z instead. So the reason why I divide X and Z into K and H and sum K and H in the form of Y is that X is the fixed effects of the linear mixed model and Z is the random effects of the linear mixed model. So ideally, they would have different common component, well, they would have different components that are not common individually, but together when they form Y, that is when they would share that information space and the latent encoding. So how do we do a transform between Y and X? For example, if Y is the histology and X is the gene expression. Well, if we write out the fixed effects of the terms in the previous slide, we get K alpha transpose for Y and K beta transpose for Y. And so the most straightforward thing to do is to convert K alpha transpose to K beta transpose. And you can do that by inverting alpha out and then attaching beta onto it after that, which occurs in the dark box below. And notice that this is a quick transform. So if X is 30,000 by, 10,000 samples by 30,000 genes, then beta would be 30,000 genes by D dimensions. And so alpha would also be 30,000 by D, but alpha transpose would be D by 30,000. And so the matrix multiplication internally is D by 30,000 by 30,000 by D. And so this matrix here is D by D. And if we choose it to be of a sufficient size that is computationally allowable for quick runtime, for example, D equals 1,000, then this here would run quite quickly. And so I show this in the next slide. For example, I start with A, I measure the time, I end at B, and in between, I extract my gene expression, I extract the alpha, I extract the beta, I convert gene expression to pixels using the expression that I mentioned earlier, and I measure the runtime. And that takes about 3 minutes for 11,000 genes on 12,000 pixels, so for 19,000 samples. So that is quite quick. And I just print the size of alpha and beta. And just notice that I set D, the dimensions, to be 1,000. And so the inverse operation here would be over the 1,000 by 1,000 matrix size. But if we were to take it in a standard linear model, it would be 12,000 by 12,000. And that is the reason why it's prohibitively large. And that implies the design model choice of this algorithm is quite efficient. So next, I show some tissue slides. On the right, we have the real histology, and on the left, we have the transformed histology. Now, this is on the training set, so we expect that the images to be almost identical because we are learning on the training set and then transforming based on the training set. I'll show the test set in a minute, but here we can see that the image is quite representative of the real histology. And likewise here for another training image. And now I'll move on to the test image where the test image is not observed in the learning of the model. And so we can see that the shape of the cells, the structure of the tissue, and the color of the tissue is similar in a way, but we don't expect it to be identical based on the fact that it's not found in the training set. And similarly here, we see that there are quite a number of cells in the real histology, and likewise, we see small faint dots in the transformed histology. So that wraps up part 2. Now, the main question is, how is this image generated? So I'll show soon that it is a linear combination of hundreds of images, and that the final image is actually a combination of the D components that I mentioned earlier. So if D equals 1,000, then it would be a combination of 1,000 images. So what I show next are the top 100 images or top 100 components that are weighted highly for this particular image I show now. So these are the top 100 components, and you can see that they vary in color, in shape, in size of cells, and even in the angular and location position of the cells. Now, it is important to note that each component is assigned to a weighted set of genes. So each image has a set of genes assigned to it in terms of weights. In fact, it's a weight across all genes. Now, because it's a weighted set of all genes, it may not directly imply certain biological phenomena, or it may not define biological characteristics as succinctly as we like. For example, if one image was related to a set of 10 genes, then that would be biologically interpretable. However, if one image is related to hundreds or thousands of genes with small weights, then that is much more difficult to interpret. So an idea here would be to regularize the weights of the genes that are assigned to each image, and that could help with interpretability. So with those 100 components, we can see here that in the top left image, we have about 30 components. But as we progressively move from one image to the next, we are adding in more components, and progressively we get to the final image, which is in the very last bottom right. And we see here that as we add in more components, we add in more information, and the shape, morphology, and structure of the image becomes more defined. So here I show a set of another examples. In the top left, I show the main transformed histology image. On the right, I show intermediate transformation of the image after about 300 components. Below it, I show progressively how adding in more components changes the image, and on the right, far right, I show the top 100 components. Likewise, I do the same for this tile and gene expression cell. And likewise, I do the same here. Now, as I mentioned earlier, each image or each component that represents an image represents a set of weights across all genes. So every component has another component in the gene space. And so if I look at the components in the gene space and then correlate them and then convert them back to the image space, these are the components, the top components I get that are correlated to this purple blotch tile here. And so you'll see here, compared to the next slide, that the cells are quite globular, they're quite rounded, diffuse. And the links between them are not, or the extracellular matrix links or shadows between the cells aren't as well defined as we see in the next slide here. So again, I correlate this yellow blotch here with other image components based in the gene space, and we see that the cells are much smaller, ovular, if there are cells, and that the extracellular matrix is a bit more defined than the previous slide. So I just re-show this again. This is the purple image, which represents the first component, and this is the yellow image, which represents the third component. Now that wraps up part 3. So I'd like to wrap it up. This is another animation of a k-nearest neighbor, representation of a histology tissue. And I'd like to now thank my collaborators, Dr. Alan Hoang, who has helped me from the beginning and has really helped me discuss some ideas about this method. And I would also like to thank these people for inspiration, especially Jeffrey McLaughlin,